In the last lesson, we started this topic on how humans impact on biological processes. We talked about this term DNA manipulation, or sometimes it's kind of called genetic engineering. And we focused on these enzymes that are used to cut, paste, and multiply DNA. Today we're going to look at these two topics. So just as a quick recap from last week, we looked at endonucleases, which are enzymes that are used to cut DNA at specific sites. We looked at ligases, which join together fragments of DNA or RNA. And then we just very briefly introduced polymerases, which we've looked at before when we've looked at gene transcription. But these are enzymes that add nucleotides to DNA or RNA and are used to copy or make copies of genetic material. So that's what we're going to focus on today. We're going to look at this particular process called the polymerase chain reaction, and then also a different process called gel electrophoresis. Now I know there's a few new big terms in here, so it's really important, especially because we're not in class and I'm not asking you questions and getting you to practice saying these words. It is really important that you do that a little bit. So it might sound a bit silly, but kind of practice talking these things, explain it to someone else, or even just say it out loud to yourself so those words stick into your mind. All right, we're going to start with the polymerase chain reaction, which is abbreviated to PCR. You do need to know what the term means, but you'll often hear of it referred to as PCR. If you want to have a quick read through your textbook, it's pages 575 to 576. You could either do that now or at the end. So PCR is a process that amplifies a sequence of DNA. What we mean by amplify is that it makes many copies. And we'll come back to that in a little bit in terms of how it works. After each cycle of PCR, and we'll look at that more in a minute, but after each cycle of PCR, we double the amount of DNA that we have. So in this picture, we start here with one fragment of DNA. We go through a cycle of PCR and we end up with two fragments. So we've copied it. And then remember, each cycle, we double that. So at the second cycle, we double this strand. So we now have two here, and we double this one. So we have two here. So we've got a total of 4. Okay, then we have 8, then we have 16. So it's an exponential growth. That's what we mean by the fact that it amplifies it. Okay, it gets bigger each time. This process allows more copies of DNA to be tested from a small original amount. So the reason we do this, or the reason scientists do this, is that a small bit of DNA can be taken, and then from that you can make thousands or millions of copies and then test each of those copies. So it's used a lot in forensics. So let's say a bit of a DNA sample was found at a crime scene, and sometimes they might just find a single hair. So they can get a small bit of DNA and then make lots of copies so that they can test it. It's used in things like paternity testing, so to test whether someone is the biological father of a child. It can be used a lot with genetic testing, so looking at disorders. So even just getting a small sample of blood, we can get lots and lots of copies of DNA from that. And you may have seen this come up in the news recently. It is what is being used for the COVID tests. Some of you may have had a COVID test by now, where they put a cotton swab up your nose and they scrape and they get some cells. From that, they extract a little bit of that DNA and then they make lots of copies of it and test to see if the viral coronavirus genome is in there. Okay, so that's some of the examples of what it's used for. Okay, it's really important that you know the steps of the polymerase chain reaction. Okay, we'll go through them in a second, but to know the steps, you need to know the materials, what is required to do this process. So firstly, this makes sense. If we want to make lots of copies of DNA, we need an original sample of that DNA. Okay, we need something to copy. That DNA, if you think back to the last lesson, is often cut out of a big section of DNA using restriction enzymes. So let's say we want to test for a genetic disease, got a chromosome, and that chromosome contains thousands of genes, and this is the actual gene that we want to test. We could use restriction enzymes to cut this gene out, and then we're going to make lots and lots of copies of that particular one. Okay. If we want to build new bits of DNA, we need the building blocks of DNA, which are nucleotides. Remember our nucleotide bases, which are adenine, guanine, thymine, and cytosine. On their nucleotides, we need those building blocks, those monomers, to create our DNA strand, which is a polymer. Remember, it's made up of individual components. We use an enzyme called Taq polymerase. Okay, so remember we've said that polymerases are enzymes that build nucleotides. Taq polymerase is the specific one that's used in this process. You do need to know that name. The reason they use this particular polymerase is that it is heat resistant. And that's really important. In a few minutes when we look at the steps of it, you'll see that in this process, it gets heated up a lot. So we need an enzyme that can handle high heats without denaturing. Okay, remember a lot of enzymes will denature at a certain temperature, their critical temperature. This can handle large temperatures, and it will basically be the machine that assembles those nucleotides to our original DNA sample. The other thing we need are called DNA primers. A primer is a short strand of nucleic acid. So they're usually about kind of 16 to 18 bases long, and they are a specific sequence, which means that they are made specific to our original strand. So the strand that we are copying, a small section will be made that will attach to the beginning of that template strand to create a starting point. When we look at the steps in a minute, that should make a bit more sense. Okay, if we look up at this picture here, we've got here our sample DNA. These red bits are the primers, and then these are our nucleotides, okay, our bases that are going to build up our copies of the DNA. All of these things, as well as our kind of Taq polymerase, which would be our enzyme, Taq polymerase, all of that will get put into a small little test tube, and they will get put into what is called a PCR cycler machine. So I know it's quite hard to see, but each of these individual little things is one of these. Okay, so they get put into this machine, and then the machine does all of the work. So these steps that we look at in a second are all happening inside this machine. So the steps of PCR, and you do need to know this, okay? This comes up in your exam all the time, and often it is a matter of describing these steps. You need to know the certain terms. The first step is called denaturation. If you remember with proteins, denaturation is when the bonds break and the protein loses its shape. It's the same principle here. In denaturation, the machine heats up to about 94 to 95 degrees, and what that does is it breaks the hydrogen bonds that are holding the two strands of DNA together. Okay, so we start with a double-stranded DNA, our template, and then these bonds get broken, and we end up with 2 template strands. Okay, we haven't copied anything yet. All we've done is separate that original strand. Step 2 is called annealing. In this step, the machine cools back down. The textbook says 55 degrees. Some videos say kind of up to 60 degrees. It's around that. I would stick with 55 degrees because that's what it says in the textbook. But the machine cools back down to 55 degrees. And what that does is it allows our primers, which remember are our short little bits of DNA, to bind into the complementary sequences on the template strands. So we're kind of just going straight from here down to this. We've got one template strand here and another template strand here, and these here are our primers. Okay, the reason it has to cool down to 55 degrees is that's a good temperature for these hydrogen bonds to reform. Okay, remember at the high temperature, the hydrogen bonds broke. So they reform and these primers attach. The primers, like I said, are built specifically for whatever this DNA sequence is. So let's imagine that we know that this is our DNA sequence of the starting bit of our gene. This primer will be complementary to that. So we'll have T, G, C, A. That will bind here, and that will basically tell Taq polymerase where to start building from. Now, hopefully you're also noticing here as well, one of these strands is going 3' to 5' this way, and one will be going the opposite way and will need the opposite primer. Okay, so that there are 2 different primers that allow both strands to be built upon. Step 3 is called elongation. Sometimes it's referred to as extension. So if you're looking up videos or other pictures, you might see it called that. Here, it goes back up to 72 degrees, and that is our optimal temperature for Taq polymerase. Remember, proteins have certain temperatures that they work best at. For Taq polymerase, that's 72 degrees. So we heat it back up, and then Taq polymerase does its job of starting at the primer, so attaching where that primer was. So we've got Taq polymerase here, and then it will one by one add a nucleotide, 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 nucleotide. So complementary to our template strand, and that will happen on both of these strands. So you notice they're building the opposite way. They always build from the 3' to the 5' of the template strand, and that will build and build and build until it gets to the end of that section of DNA. And then finally, we repeat this cycle. Okay, so the cycle technically is steps 1, 2, and 3, and that will get repeated until we have as many copies of the DNA as we originally, as we need. Okay, so you really need to know these steps in their correct order and what happens. So denaturation, annealing, and elongation, and then we keep repeating that until we've got as much DNA as we need. Just a couple of other pictures that show this in order. So this is our template strand. So technically, actually, that's not a process yet. This is our beginning template strand. Step 1, denaturation. We separate those strands. That's at our magical temperature of kind of 94, 95 degrees Celsius. The hydrogen bonds are broken. Number 2 is annealing. So we go back down to about 55 degrees. The primers join on both of those strands. And then 3, we have our elongation. We go back to 72 degrees, and Taq polymerase binds and starts building those template strands. okay? And then we can repeat that cycle. So that is now showing that we've separated these two strands and these two strands. So now we're making we've got four templates, and we're going to make 8 copies. Then we could do the same with each of those, okay? This is a different picture that shows it with all the kind of materials and then the steps here. It's really up to you. You should definitely have a picture in your notes that show this. It just depends what picture sits best with you. And there is one more kind of showing the same thing. Okay, as you can see, really important that you know those terms. Right, what I'd like you to do now, before we move on to the next little topic, I've posted a link to a really short kind of 2-minute video. The videos for this are not, you know, the most amazing, exciting quality, but they will help you to visualize it. So watch this. Feel free to Google and try and find some other ones if you like. I found a few that relate to the COVID testing, but if you watch that, just note that there's some other steps that happen in that one before it gets to the DNA PCR bit, so it is a bit more complex. So watch that video, and then I want you to go into Edrolo online, answer as part of 15B, questions 1 to 7. Just note you don't need to do question 4. Question 4 relates PCR with a different process that has been removed from this year's study design. So don't worry about question 4, but do 1, 2, 3, 5, 6, and 7, and then come back here and we'll move on to the next bit. All right, so the next section is just following on in the textbook, pages 581 to 583, and this is called gel electrophoresis. This is a process that allows scientists to separate fragments of DNA based on their size. Okay, so imagine here we've got 3 different genes, 1, 2, and 3, and we want to find out which one is longest and which one is shortest, or we want to separate them based on their size. We'll come back to some of these terms in a minute, but you can see this first one here, we don't know this yet, but it's 500 base pairs long. This one is 800 base pairs long, and the third one is 700 base pairs long. So basically, all you need to know is you've got 3 different bits of DNA. We want to separate them. This is usually used after either we've cut DNA from a larger section of DNA using our restriction enzymes, or we have just amplified bunches of DNA using PCR, and we want to kind of separate them. You might be thinking, well, PCR makes exact copies. It does, except the first few cycles, I think it's the first 3 cycles, produce DNA with variable length. That's to do with the primers and the fact that it takes a few cycles to get the exact gene length. Don't worry too much about that. It's quite complex, but just note that in PCR you do get a few pieces at the beginning that are longer than what we actually want. To do gel electrophoresis, DNA fragments are placed in a gel bath and an electrical current is applied to that bath. This is a picture of what it would look like in a lab, so it's kind of a bit blurry, but these are the hands of a scientist. This here is a gel bath. They're looking at the results, and all of these little bits are bits of DNA. Okay, usually when you get it in a saccharin exam, it will look a bit different. We'll go through that in a few minutes, but that's what it will actually look like in a lab. We'll come back to this in a second, but it's important to know that the smaller fragments of DNA move faster through this gel than the larger fragments. Again, we'll come back. So the setup of gel electrophoresis, you do need to know how it is done. So we have this bath which contains a gel. In the picture here, that is the grey component. You should either be drawing this in your notes, or you can take this picture from my slides and make sure you label it. But there's a gel in there. The gel has little pores in it, little holes, which is important because it allows the DNA to move through the gel. On top, we have this buffer solution. So it's basically just a liquid that is placed on top of the gel. It's important because it allows an electrical current to be passed through the gel. We have a negative electrode. The position of that is really important. I'll come back to that in a second. But up here we'll say negative electrode, and then we have a positive electrode. Okay. And then also there would be a power source. So both of these would be linked to an electricity source. We have these little things up here called wells. Okay. And they're basically a little indent. This is where the DNA will be placed. Okay, so the DNA that we want to separate will get placed in these wells. The important thing you need to know is that the wells are on the same end as the negative electrode. Okay, always on the same end as the negative electrode. You could get a question in an exam that showed you the picture of the wells and asked which side would the negative electrode be placed on. Also worth noting, if you remember in the last picture, it kind of looked a bit fluorescent. That's because DNA itself is actually invisible. So to see the results of this, the gel that we put in at the very beginning is dyed in a fluorescent dye. And after the whole thing has been done, it'll be looked at under a UV light, which is how you can actually end up seeing the DNA. Okay, so this is how we set it up. We've got DNA going into these wells, we've got this gel, and we've got an electric current that is going to pass through it. The theory on how this works, or why this works, is that DNA is negatively charged. All DNA, it doesn't matter what the combination of bases are, all DNA has a negative charge. So if we put something that is negatively charged next to a negative electrode, it will get repelled from it. Okay, so it's kind of like when you put the same end of two magnets together and they want to push away from each other. The negatively charged DNA will be attracted to the positive electrode up here. So the DNA will move towards the positive electrode. And you need to know both of these in terms of the theory. Smaller bits of DNA will move easier and faster. Okay, so if we put a bunch of DNA up here and they're all different sizes and we turn on a charge, they will all move, but smaller pieces will move faster than bigger pieces. So if we put a current on for an hour and then we turn it off and we see where those bits of DNA are, we can say, well, this one is smaller than this one because it moved further in the same amount of time. This is, you could definitely get a question that asks about how gel electrophoresis is set up. So you do need to know that last information, but you definitely need to know this as well. How to read the results of a gel run is what it's often called, of DNA. So as we said, DNA gets placed in these wells. Okay, so let's say we're looking at 3 different people. We'll call them samples, sample A, B, and C. Each of these wells will get multiple pieces of DNA. You don't just put one piece of DNA in there. There might be thousands of pieces of DNA. The important thing is that they all come from the same sample. They're all coming from the same person in each well. The current is switched on. So remember, we've got the negative here, we've got the positive here. When the current is switched on, the DNA is going to start moving down here. The smaller bits will move faster. When we turn off the current, the DNA will have moved and will settle in bands. Okay, so the gel is quite thick. So once the current is off, the DNA won't move anymore. So it'll settle into bands of the same length. Okay. I've added in now some diagrams here. So some labels. So let's say we've got fragment 1, fragment 2, 3, 4, 5, and 6. Have a look for a second and see if you could make comparisons between each of these fragments based on what we have just said. So let's start with fragment 1 versus fragment 2. So if we were to compare this fragment, which is fragment 1, and fragment 2, they're both in the same place. They've both moved the same distance in the same amount of time. So we could say fragment 1 is the same length as fragment 2. Okay, if you want to pause quickly and look at the other ones and just check in a second. So now we'll go fragment 3 versus fragment 4. So fragment 3 has not moved as far as fragment 4, which means that it is bigger. It's slower to move. So fragment 3 is bigger than fragment 4. Fragment 4 could move faster because it's smaller. And finally, fragment 6 compared to fragment 5. So fragment 6 has moved the furthest in the same amount of time. It's moved much further than fragment 5. So fragment 6 is smaller than fragment 5. Okay, so we can compare the different sizes of the DNA based on how far they've moved. But does this tell us how long each fragment is? Remember at the start when I showed 3 different genes, I think it was 500, 800, and 700 base pairs. This, just as we've got here, doesn't tell us how long they are. It just says which one's smaller and which one's bigger. If we want to know the actual length of the fragments, and usually we do, these things called ladders or standard ladders or markers are used. Okay, so we've got the same thing here. We've still got the same sample A, B, and C, but also in the first well, and it's usually in well number 1, we place these markers or ladders. Okay, a ladder is a fragment of DNA with a known length. We know how long these fragments are. And the way that is measured is either as BP, which means base pairs. So you can see here we've got 1,000 base pairs, 900 base pairs, 800 base pairs, so on and so forth. Or if they're really big sections of DNA, they might be measured as KBP, which is 1,000 base pairs. So up here, basically, that would be 1 KBP. Okay? So in this first well here, in well number 1, there would have been lots of different DNA fragments placed in there. Some of them were 1,000 base pairs long, some were 900, some were 800, some were 700, 600, 500, 400, 300, 200, 100. Okay, so they were all placed in there at the beginning, along with sample A and B and C, and the whole thing was turned on for a couple of hours and turned off. We know these are going to go out into this kind of format because we know how long they are. So what we're using this lane for is to estimate how long the fragments in the samples are. Okay, so again, we've numbered 1, 2, 3, 4, 5, and 6. Pause for a second. See if you could estimate the length, the exact base pair length of each of these fragments. Okay, so fragment 1, which hopefully you notice is the same length as fragment 2, is 1,000 base pairs long. Okay, make sure you put in the measurement that it's given you. Fragment 4 is about the same as 800 base pairs long. Fragment 5, so this is between two of them. It's between 500 and 400, and it's a bit closer to 500. So it's probably about 460 base pairs long. Okay, and fragment 6 has gone past 100 base pairs, so we could say it's less than 100 base pairs long. We don't have the next comparison, so we're not completely sure. It's probably around 80 base pairs long. Okay, that is a common way of how you would be assessed this in your SAC or in your exam. In the Ed Rollo questions, there are a few examples. Okay, so for that section, jump into Ed Rollo questions 1 to 6 of 5C. And then once you have done all of that, I just want you to do my Google quiz and feedback. Once I've got the feedback, I did receive all your feedback from the first lesson. If there's a few things that people want to go over, we'll look at doing a Zoom in our next session just for about 20 or 30 minutes just to go through some of those topics again. Thank you.